So the function of the different neuroglial cells. Let's take a look real quick. This will actually be better than me just talking about because you can actually see their pictures up here. How many types of neuroglial cells are there? Six. All right. So let's just take a little review of those six. And you may have noticed that some of them have the same function. They're just in different places. Whichever, it doesn't really matter so much about where they are. I just want you to know what each of them does. So we've got our astrocytes, and they basically make sure that the environment around the neuron is correct. So like the right amount of ions, kind of protect them from toxins. That's the astrocytes. And then we have our oligodendrocytes. They make the myelin sheath, which is the same as, who else makes myelin sheath? What other cell? That's also the Schwann cell there. So oligodendrocytes and Schwann cells that make the myelin sheath. That's that special kind of covering that helps with signal transmission. And then we have our microglia, and they're actually a type of phagocyte. So they're responsible for eating any foreign invaders. So you can see them up here, and in that little picture you can see they're eating something. A little hungry white blood cell. And then ependymal cell, they actually make the cerebrospinal fluid. That's their main function. And then satellite cells do the same thing as astrocytes. So they're going to maintain the chemical environment, just sort of general protection. So keep toxins out and make sure it's got the right amount of calcium ions, sodium ions, everything like that. Right. What else? Anybody? Last chance, last call. Say it one more time. Describe the three. We'll go over that one today. You don't need to know it for the quiz. It's pretty complicated, I think. All right. Let's go ahead and take the quiz then. There we go. So for the first one, what is part of the central nervous system? Is it brain, neurons in the muscle, spinal cord, or A and C? It's A and C, exactly. That's your central nervous system. Which of these is not a type of neuroglial cell? Schwann, astrocyte, neuron, or satellite cell? It's the neuron. The neuroglial cells support the neurons. The neurons are like their own sort of category. And what is a synapse? Part of the brain, place where two neurons meet, a disease, or none of the above? So it's a place where two neurons meet. Are synapses only in your brain? They're not. They're all over the place. I think a lot of times we hear synapse and we just think brain, because that's the most, where you hear most about them, but you have synapses all over because your neurons meet each other. And which neuroglial cell makes the myelin sheath? Was it Schwann, astrocyte, neuron, or satellite cell? Schwann. It is the Schwann cell. And what is a ganglia? Type of neuroglial cell, a cluster of neuron cell bodies, or a group of axons? It's a cluster of neuron cell bodies. Right. New notes. All right, so this chapter is probably the hardest one for this unit, mostly because it's the most microscopic. So if you had any troubles at all, kind of with how muscle tissue works, how muscle cells work, how nervous tissue work is also going to require some extra time and effort probably. And then once we get to the brain and those things, they're a little easier than kind of how it works at the basic level. But if you don't know how it works at a one cell level, the whole rest of it isn't really going to make any sense. So that's why we have 2 days for this chapter, is to make sure you can zoom in all the way and know what's going on. And you may see a lot of similarities between muscle cells. So like, what was the signal that made muscle cells contract? Do it? So an action potential, that's also going to come up with these neurons, that they're activated by action potentials, and that's how they send signals down their axons. But first, we're going to talk a little bit about what they look like, and midbrain potentials. And next time, we'll talk more about how the signals are transmitted down the neurons. So we looked at those neuroglial cells, and we could take a look at the neuron itself. So one thing to remember about neurons is they have 2 different ends to them. And so at one end, you actually have the dendrites, which are these little sensory things on the ends. This is where signals are received or sensed. And that's attached directly to the cell body, which is the nucleus. So you can see that little blue nucleus in there. And then the long, long strip that the signals travel through, which can be, anybody know what the longest neuron is? Like the length? Any guesses? It is definitely more than 3 centimeters. It's about 3 feet, depending on how tall you are. So that's a neuron that goes from like your toes all the way up to your spinal cord. So you have that's one cell that goes that far. Which means this axon here, which is the really long transmissible segment, that's the part that's so long. It's like 3 feet long, which is pretty wild when you think about how tiny everything is in your body, right? So the signals travel from dendrites to cell bodies, down the axons, and then down here is your axon terminal. Any guesses what these little sausages around there are? Close. It's the cell that makes the myelin sheath. So that'd be the Schwann cell. So the Schwann cell is like in orange or red there, and then that yellow thing around the axon is the myelin sheath itself. So the cell's, around it, and then it puts myelin sheath inside. And then the dark yellow is the actual axon of the neuron. So that's what they look like. Same picture, and as we'll see in a minute, they are electrically excitable, which means when they get excited or activated, the voltage changes. And so the way they transmit signals is through actually letting in positive ions So like a voltage change, and that's going to starts in the dendrites, and then it goes all the way down the axon to the axon terminal. And so you can think of like it starts negative inside, and then it's going to become positive, and that positive ion is going to travel as an electrical signal all the way down to the bottom. But we'll talk more about that next time. So the dendrite, that's the part that senses. So it can either, maybe it's sensing temperature or pain or touch. And then if it gets activated, it sends a signal down the axon, and then it eventually gets to axon terminal. And then the axon terminal actually releases a signal like a neurotransmitter, like you saw with those muscle cells, where you make a little chemical and it goes outside the cell to bind to another cell to tell it to do something. And then, of course, we have our Schwann cell, and then the myelin sheath. It's like basically acts as an insulator for electricity so that the signal can travel faster. Just like, you know, how you have insulation around wires to make them be able to conduct electricity. Same thing with those neurons. And one interesting note to make, those neuroglial cells, obviously they're not neurons, so they don't get excited by electricity or do anything with ions like that. And one thing they can do is multiply and divide. So can your neurons multiply and divide? Except in rare cases, they cannot. But what can is all those cells that support the neurons. So if some cells die that are making the myelin sheath, you just make new ones. Or if some of those white blood cells that are living in there, if they die off, it's okay, you just make new ones. But you can't make new neurons. So if that neuron is completely damaged and dead, that's it. Which is why, paralysis isn't so repairable. There are disorders where, like, some of these neuroglial cells will become damaged from some disease or trauma or something, and you'll lose the ability to transmit signals, but then these cells can grow back, and then you'll regain that ability if the cells aren't, if the neuron wasn't actually damaged. It's kind of interesting. All right. So question 1, the dendrites of a neuron are blank. All right, final answers. Let's see what you got. So everybody changed to A, which is correct. So they're the receiving region of a neuron, which means they don't secrete neurotransmitters. They receive them instead of secrete them. And they don't really conduct impulses. That would be the axon that conducts impulses toward another one. And they do carry action potentials, as you'll see in a moment. All right. So the first thing we're going to do is draw a neuron without looking at one to make sure you got it. So maybe in pairs makes the most sense. So it's question 1. All right, so nice work on those. Just a quick double check, just to remind you. The axon is a long tube that goes through the middle, and then outside of the axon is the myelin sheath, then outside of the myelin sheath is the Schwann cell. So it's like a 3-layer sausage or something, I don't know. So you've got the inner piece, and then you've got, the mustard, And then you got your bun on the outside. That's how it works. And it's one super long sausage and a bunch of individual little buns around it. If that makes sense. You can tell I'm a little hungry. Crazy. When I lived in Poland for a little bit, for a year teaching English, there's actually sausage stores, and you'd go in and all it would be is just the whole wall of just all sausages just hanging on a wall. Most of which looked disgusting, but yeah, I know. All right, so are you good on the structure then? Ready for something harder? All right, so since neurons are electrically excitable, that means they have very special ion channels that keep ions in one place versus another, just like your muscle cells. And membrane potentials are very important as well. And ion channels maintain membrane potentials. So first off, make sure you're getting what the different channels are. So all the ones we're going to talk about, there's special holes in the cell membrane to let ions through. Because can ions go through cell membranes by themselves? They can't. Why not? No, don't say they're too big. They're too charged. There you go. They're tiny. They're tiny, tiny, but they are charged, which is good because then the cell can decide which ions are where. And in order to do that, you've got all these special channels that are little holes in the membrane that can let ions through, and they're specific. So we've got 4 main kinds, and what's different about them is what tells them to open or close. So the first one is a leakage channel, and this type of channel just randomly opens and closes. It's under no control of the cell itself. It's just open 50% of the time, close 50% of the time. So those are leakage channels. One kind you have is a potassium leakage channel. And sometimes it's open and it lets potassium out. Sometimes it's closed, and then potassium can't get out. And kind of makes sense why it's called a leakage channel, right? It's like something you can't control. It's just a leak. It's kind of the idea. Another one that you actually may have seen already is the ligand-gated. So this one is one where a neurotransmitter or some molecule outside the cell binds to it, and then it opens it. It's like somebody puts a key in it and it opens. If there's no ligand over here, then it's closed. The ligand's this little orange thing. It's not touching it yet, so it's closed. When it touches it, it opens it. So a molecule, really commonly would be a neurotransmitter. Mm-hmm. Do you remember seeing some kind of ligand-gated channel when we studied the muscles? You may remember?
Yeah, the calcium. Is it calcium that?
That would have been one. What about one that was on the cell membrane, though? Yeah. Do you remember what happened when acetylcholine bound to those receptors? They opened and they let sodium in. So that was an example of a ligand-gated channel. And just like all the other ones, each of these channels, this one would be specific to some certain ion. Let certain things in or out. One that is very unique is a mechanically gated channel. So the way this one works, so right now if you just hold your hands out, is there anything touching your fingers right now? What if you push on something? Do you feel any pressure? So the reason you feel pressure is because you have neurons in your fingers that whenever they feel pressure, they open. So you pushed on there, this channel felt the pressure and it opened, let in some ions, and then it started a signal. So those are what's called mechanically gated channels. Pretty crazy. And then the last one, and most confusing, is the voltage gated channel. So your neurons also have voltage gated channels, and they're only open at certain voltages. Which means, for example, on this one, when we say the voltage is minus 70, does that mean the inside of the cell is more negative or more positive than outside? It's more negative. So whenever the inside of the cell is more negative, like minus 70 more negative, this channel's closed. Whenever the cell becomes more positive, like minus 50, then the channel opens and lets in the ions, or lets ions in or out. That's the voltage gated one. All right, so I'm going to have you do a couple Venn diagrams to compare and contrast those two, or those four, I suppose. So see what you can come up with there. All right, so let's go over those and make sure everybody's got everything on there. So ligand versus voltage gated. What was the difference? So chemical stimulus opens it. And then what about the voltage one? So like voltage change. Some people wrote membrane potential change. That's the same thing. What'd you find for both? So they are ion channels. Where would you put specific to certain ions? Both voltage or ligand, specific to certain ions. So it's actually both. So they're both specific. They're not just random holes and everything can come in and out. They have a certain shape that says, okay, I'm this one channel, I'll let potassium through. Or I'm this one channel, I'll let calcium through. So they are specific. Anyone have anything else? Yeah, that's a good one. That's not, we're going to talk more about that next time. But yeah, the ligand ones are in the dendrite usually. And then the voltage are like in the axons. Or at least usually. All right. What about mechanical versus leakage? What's something that's true just of leakage? Random. And then what opens a mechanical one? pressure sensitive. Right, and what about both? Okay, they're both ion channels. What else?
Is the weighted one ion specific?
Just like we said over here, they're also both specific to certain ions. Right. And I think that's plenty for those. So if you notice, what do all of them have in common? They're all ion channels, and they're all specific to certain ions. And then what's different is what makes them open. It's a thing to remember. All right, so your neurons, when they're not sending signals, are activated, just like your muscle cells, when they're not contracting. They have more negative charges inside than outside. That's called a resting membrane potential, and because it's more negative, we'd call it a negative. And it's pretty significant. It's minus 70 millivolts more negative inside than outside. Let me write. All right, so it's kind of interesting how it gets to be this way. So if this is our cell, then this is outside. And there'll be better pictures in the PowerPoint too, but I'll try and draw it too. Inside the cell, obviously, you've got more proteins inside your cell than outside, because, I mean, just kind of makes sense, right? The cell's making the proteins, so they're in there. And proteins tend to be negatively charged. So we've got lots of proteins that are negative. And then phosphate ions. Does anyone know two things in your cell that have lots of phosphates? No. Maybe some. What's the energy of the cell? What does mitochondria make?
ATP.
What does P stand for? So ATP has 3 phosphates. Phosphates are very negative. So that's one place. So we've got ATP that's negative. Is there what's in the nucleus? What molecule? So DNA is the nucleus, and every nucleotide has a phosphate group as well. So your DNA is very negatively charged. So DNA is negative. So those are the main places you get phosphate ions from. So those 3 things make it more negative. And there is more potassium inside, which is positive. So it kind of cancels out some of the negativeness. So you'll see. And then outside the cell, there's lots and lots of sodium. And then some chlorine. So if you notice, we've got, this one, the proteins, ATP and DNA, that are all making the inside more negative. And then there's lots and lots of sodium, which is positive outside. So these 3 compared to this one is what's mainly giving it that negative charge. So it's a little confusing because there is stuff that's positive inside. It's not like there's nothing. And there's stuff outside that's negative. So it's not like there's nothing negative outside. But if you add them all together, it's a lot more negatively charged stuff in there than the outside. Which is important because how do you think we're going to trigger the cell to get active. So it's just like the muscle cell. We're actually eventually going to let this sodium come in, and that's going to be the signal to start everything. But that's next time, probably. All right. We also, so we talked about those. And some of the ways that the cell maintains these concentrations are having lots of K plus channels and not very many Na plus channels. So, we take a look at this one. Does anyone see potassium up there? It's not labeled. Oh. They're the little orange triangles here. So do ions move from low to high or high to low? They go from high to low. So we've got lots of potassium inside, not much outside. So which way is potassium going to go? So it's going out, and if we notice we've got this leakage channel here, is that controlled or is that just randomly open? It's randomly open. So we've got more potassium, and it's leaving. So if you lose positive charges inside, does that make your membrane potential negative or positive? If you're losing positive from inside, makes it more negative. So we've got a decent amount of those. So lots more potassium leak channels, and then we've got a few sodium leak channels. So that's sodium, because it's higher outside. Sodium comes in. What does that do to the membrane potential? It's more positive, right? Some positive ions in. So if you've got more potassium than sodium channels, what's the net result? More negative or more positive? More negative, right? Because we've got more potassium leaving than sodium coming in, just because there's more holes for it. So they're specific. Obviously, since these are leakage channels, what would happen if that's all you had? What would happen to the potassium ions? They'd all be leaving, right? And then all the sodium would come in, and your membrane potential would suddenly not be there. So in order to keep everything sort of stable, we've got our sodium-potassium pump. And if you take a look at what it does, it pumps 3 sodiums out and 2 potassiums in, which keeps the potassium in, the sodium out. And also, if you pump 3 sodiums out and 2 potassiums in, are you making it more negative or more positive inside? More negative, right? Because we pumped 3 out and 2 in. So that's like pumping 1 extra positive out, right? So those things all together keep things more negative inside. So you've got lots of your proteins, the phosphates, and then basically pumping the sodium out and the potassium in makes it more negative as well. All right, so let's switch these. Is that a higher concentration inside the cell than outside? So it is E. Which ones? Oh, sorry, F. It is F. So I get confused how many letters I have up there sometimes. So C is true. E is true, and then there's one more. That is A. So A, C, and E are all true. Right. All right, so we've got plenty of time, so why don't we take, I don't know, 4 minutes just totally on your own to answer any of these on the back side that you think you can. So we want to know, would the resting membrane potential become more positive or more negative? And explain. So we'll give you 4 minutes just on your own, and then we'll let you work together. All right, let's take a look at a quick little animation. Then I'll give you a chance to work together, and maybe this will help clear things up a little bit. So this is basically about how you keep the membrane negative inside.
Key factors. First, an unequal distribution of ions and charges across the membrane. Second, a greater permeability of the membrane to potassium ions than sodium ions because of the presence of a greater number of potassium pores. This results in more positive ions leaving the cytosol and making the inside slightly more negative. And finally, the effect of the sodium-potassium pump.
Over here.
Because some sodium is always leaking in and potassium is leaking out, the pump helps by pumping out sodium as fast as it leaks in, thereby maintaining the diffusion gradient of these ions. The result is a constantly maintained electrical charge across the membrane.
And if you saw that pump, pumped 3 out sodiums, 2 potassiums in, which is like a net -1, right, for every time it goes. All right, so compare with your group there for that question. I'll go over it. In that picture, outside is up. All right, so let's go over those. Make sure you got them. So for A, increased sodium leak channels. Would it make it more negative or more positive? More positive. Sodium's normally, higher outside. It's positive. It comes in, right? So more positive. More potassium leak channels. More negative because the positive ions are leaving. Fewer sodium-potassium pumps. Be more positive. Because sodium-potassium pumps, they, each time they pump, it's 3 positives out, 2 positives in, which makes the inside more negative. So if you have less of them, you'd be more positive. 3 positives out, 2 positives in, that's a net of -1. So then D, what's the answer for D then? Be more negative, because now you've got more of them, so more positives out. And what about E? So what if suddenly we put a bunch of big holes in here and everything could move from high to low? So it would become, if it started -70, what would happen to the sodium? So sodium's going to come in. Potassium's going to go out. I didn't say how big the holes are, so do you think maybe the proteins are leaving too? Could be. ATP's leaving. The chlorine's coming in. So it basically becomes as if you didn't have a membrane here. Everything would diffuse from high to low, and eventually you'd get to 0, right? You wouldn't have any sort of positive or negative amounts. So, if you say you start at -70 and you go to 0, you'd be getting more positive and eventually getting to neutral. So that would be like a really bad poison or something. And for all those, we were starting at -70. That's where the cell would normally be at. So interesting enough, so you just did these 4 things where you changed the amount of channels. That's how you can change the membrane potential. So for example, one interesting one, have you heard of pacemaker cells in your heart before? Maybe. So there's certain cells up in the heart that actually slowly let in sodium ions at a steady rate. And once they let in enough sodium ions, then they signal contraction, and they tell the whole heart to contract. So they actually have, if you had to guess, what do you think they have a lot of? If they tend to have positive ions coming in somewhat quickly. So they have a lot of sodium leak channels, more than like your regular neurons do, because they're a pacemaker. So what they want to happen is sodium to just come in pretty quickly, and then signal contraction, signal the signal to start and everything, without anything having to tell it. So, right. Yeah, and then to change your membrane potentials, you change the channels. That's a good question. So for example, your muscle cells, do they spontaneously contract without you telling them to? Sorry, your skeletal muscle? It doesn't. So that means they don't have very many sodium leak channels because they don't just spontaneously start contracting all of a sudden, which would be bad.
They do, that's called a cramp.
Do it?
So they do, but that's called a cramp.
Yeah. So anyways, we've got 2 more minutes. Just wanted to tell you while we're talking about it is how you at least start a neuron to be activated is something happens at the dendrite to open sodium channels. If you open a bunch of sodium channels, what happens to the membrane potential? Becomes more positive. So you might have sensory receptors, like those mechanically gated ones. Press on them, they open, sodium comes in. Now the inside of the cell is more positive. Or maybe it's a ligand-gated channel. So a neurotransmitter binds to it, it opens, it lets sodium in. Inside the membrane becomes more positive. Or maybe it's a voltage-sensitive one, where you change the voltage at the dendrite, and it opens those channels and lets sodium in. So what we're going to be talking about next time is how all that works. But they do all start the same way. Basically, a channel gets signaled to open, and it lets sodium in. So that's it for today.